DNA replication, repair, and recombination, ChatGPT said. DNA replication, repair, and recombination. DNA replication, repair, and recombination are essential processes ensuring the stability and integrity of the genetic material in all living organisms. These mechanisms allow cells to accurately duplicate their genomes, correct errors, and facilitate genetic diversity. 1. DNA replication 1.1. Overview. DNA replication is a semi-conservative process in which each parental DNA strand serves as a template for the synthesis of a new complementary strand. This process occurs in the S phase of the cell cycle and ensures the accurate transmission of genetic information. 1.2. Steps of DNA replication 1.2.1. Initiation. Origin of replication or DNA replication begins at specific sequences called origins of replication. Initiator proteins. In prokaryotes, DNA A binds to ORC, initiating strand separation. In eukaryotes, the origin recognition complex ORC recruits helicase. Helicase DNA B in prokaryotes, MCM in eukaryotes unwinds the DNA double helix. Single-strand binding proteins SSBs prevent reannealing of separated strands. Topoisomerase relieves supercoiling stress ahead of the replication fork. 1.2.2. Elongation. Primase synthesizes short RNA primers to provide a 3' group for DNA polymerase. DNA polymerase. Prokaryotes, DNA polymerase 3 is the primary enzyme for elongation. Eukaryotes, DNA polymerase alpha primase activity delta lagging strand synthesis and epsilon leading strand synthesis. Leading strand synthesis, continuous synthesis in the 5' right pointing arrow 3 direction. Lagging strand synthesis, discontinuous synthesis through Okazaki fragments, later joined by DNA ligase. Sliding clamp beta clamp in prokaryotes, PCNA in eukaryotes enhances polymerase processivity. 1.2.3. Termination. Prokaryotes, replication ends at tear sequences, recognized by the 2S protein, preventing helicase movement. Eukaryotes, ends of linear chromosomes require telomerase, a reverse transcriptase that extends telomeres to prevent loss of genetic material. 2. DNA repair mechanisms. DNA is constantly subjected to damage from replication errors, chemical modifications, and external factors UV, radiation. Cells have evolved repair mechanisms to maintain genome integrity. 2.1. Direct repair. Photoreactivation. UV-induced thymine dimers are repaired by DNA photolysis prokaryotes and some eukaryotes. 0,6-methylguanine DNA methyltransferase MGMT repairs alkylated guanine. 2.2. Excision repair. Base excision repair BER. Repairs small, non-helix distorting base modifications e.g., deaminated cytosine right pointing arrow uracil. DNA glycosylase removes the damaged base, AP endonuclease cuts the backbone, and DNA polymerase beta eukaryotes or I prokaryotes fills the gap. Nucleotide excision repair NER. Repairs bulky lesions e.g., pyrimidine dimers, chemical adducts. UVRABC endonuclease prokaryotes and XPC, XPA, XPB, XPD eukaryotes recognize and excise damaged nucleotides. DNA polymerase fills the gap. 2.3. Mismatch repair MMR. Corrects replication errors, mismatched bases, small indels. Prokaryotes, MUTS recognition, MUDL recruitment and MUF endonuclease. Eukaryotes, MLH1, MSH2, PMS2. Mutations in MMR genes lead to Lynch syndrome, hereditary non-polyposis colorectal cancer, HNPCC. 2.4. Double-strand break DSB repair. Homologous recombination HR error-free repair using a homologous sequence as a template. Uses BRCA1, BRCA2, RAD51 in eukaryotes. Non-homologous end joining NHEJ error-prone repair that directly ligates broken ends. Uses Q70, Q80, DNA PKCs. 2.5. Translesion synthesis TLS. Specialized polymerases e.g., DNA pol eta, zeta bypass DNA lesions at the cost of introducing mutations. 3. DNA recombination. 3.1. Homologous recombination HR. Occurs during meiosis, mitosis DSB repair, and horizontal gene transfer. Key steps. Strand invasion, RECA prokaryotes, RAD51 eukaryotes. Holiday junction formation and resolution. 3.2. Site-specific recombination mediated by recombinases e.g., CRE, lambda integrase. Example, integration of bacteriophage lambda into the E. coli genome. 3.3. Transposition. Transposable elements, jumping genes, move within genomes via transposase. Types. Class 1 retrotransposons, RNA intermediates, e.g., lines, signs. Class 2 DNA transposons, direct DNA movement, e.g., is elements, P elements. Practice questions with answers. Previous year question, gave ecology and evolution Q1. Which of the following is the most error-prone DNA repair mechanism? A. Homologous recombination. B. Base excision repair. C. Non-homologous end joining. D. Nucleotide excision repair. Answer. C. Non-homologous end joining. NHEJ directly joins broken DNA ends without a template, often introducing mutations. Multiple choice questions MCQs. Q2. Which enzyme is responsible for removing RNA primers in eukaryotic DNA replication? A. DNA pol 3. B. DNA pol alpha. C. DNA pol delta. D. RNase H. Answer. D. RNase H. RNase H removes RNA primers, and DNA pol delta fills the gaps. Q3. Which protein is primarily responsible for strand invasion during homologous recombination in eukaryotes? A. RECA. B. RAD51. C. BRCA1. MUTS. Answer. B. RAD51. RAD51 facilitates strand invasion by pairing homologous DNA sequences. This detailed coverage of DNA replication, repair, and recombination aligns with PhD entrance preparation standards and includes previous year questions and MCQs for effective revision. Let me know if you need further elaboration. You said. Unit of replication, enzymes involved, replication origin and replication fork, fidelity of replication, ChatGPT said. DNA replication, unit of replication, enzymes, replication origin, replication fork, and fidelity. DNA replication is a highly regulated process ensuring the accurate duplication of genetic material. This section covers key aspects such as the unit of replication, enzymes involved, replication origin, replication fork, and the fidelity of replication. 1. Unit of replication. The unit of replication, or replicon, is a segment of DNA that is replicated from a single origin of replication. Prokaryotes e.g., E. coli. Have a single replicon due to a single origin of replication or EC on their circular chromosome. Eukaryotes, e.g., humans. Have multiple replicons with multiple origins of replication to accommodate large linear chromosomes. Each replicon functions independently, ensuring the entire genome is replicated efficiently. 2. Enzymes involved in DNA replication. 2.1. Helicase. Unwinds the DNA double helix by breaking hydrogen bonds between base pairs. Prokaryotes, DNA B, part of the chromosome complex. Eukaryotes, minichromosome maintenance, MCM, complex, MCM 2-7. 2.2. Single-strand binding proteins, SSBs, prevent reannealing of separated DNA strands. Prokaryotes, SSB protein. Eukaryotes, replication protein A, RPA. 2.3. Topoisomerase. Relieves supercoiling caused by helicase activity. Topoisomerase I, introduces single-strand breaks. Topoisomerase II, e.g., DNA gyrase in prokaryotes introduces double-strand breaks to relieve torsional strain. 2.4. Primase. Synthesizes RNA primers to provide a free 3' group for DNA polymerase. Prokaryotes, DNA G primase. Eukaryotes, DNA polymerase alpha has primase activity. 2.5. DNA polymerases. Prokaryotic DNA polymerases. DNA pol I, removes RNA primers and fills gaps. DNA pol 3, main polymerase for elongation. DNA pol 2, IV, B, involved in DNA repair. Eukaryotic DNA polymerases. DNA pol alpha, initiates replication by adding RNA primers. DNA pol delta, synthesizes the lagging strand. DNA pol epsilon, synthesizes the leading strand. DNA pol beta, involved in DNA repair. 2.6. Sliding clamp. Enhances polymerase processivity, ability to add nucleotides continuously. Prokaryotes, beta clamp part of the DNA pol 3 holo enzyme. Eukaryotes, proliferating cell nuclear antigen, pCNA. 2.7. Clamp loader. Loads the sliding clamp onto DNA. Prokaryotes, gamma complex part of DNA pol 3 holo enzyme. Eukaryotes, RFC, replication factor C. 2.8. RNase H and DNA ligase RNase H, removes RNA primers in eukaryotes. DNA ligase, seals nicks in DNA by forming phosphodiester bonds. Prokaryotic DNA ligase, uses NAD+. Eukaryotic DNA ligase, uses ATP. 3. Replication origin and replication fork 3.1. Replication origin. The origin of replication ORI is a specific DNA sequence where replication begins. Prokaryotes. Single origin ORI-C in E. coli. ORI-C contains DNA A boxes where DNA A binds to initiate replication. Eukaryotes. Multiple origins ARS, autonomous replication sequences in yeast. Origin recognition complex ORC binds to origins and recruits replication proteins. 3.2. Replication fork. The replication fork is a Y-shaped structure where active DNA synthesis occurs. Key components of the replication fork. Leading strand, synthesized continuously in the 5' right pointing arrow 3 direction. Lagging strand, synthesized discontinuously as Okazaki fragments. Replication proteins helicase, primase, DNA polymerases, etc. Function at the fork, bidirectional replication, DNA synthesis proceeds in both directions from the origin. 4. Fidelity of DNA replication. DNA replication is highly accurate, with an error rate of 1 in 10 to the power of 9 10 nucleotides due to multiple proofreading and repair mechanisms. 4.1. Proofreading by DNA polymerase. DNA polymerases have 3' right pointing arrow 5, exonuclease activity, allowing them to remove incorrect nucleotides. Prokaryotes, DNA pol 3 has proofreading ability. Eukaryotes, DNA pol delta and epsilon have proofreading functions. 4.2. Mismatch repair MMR, system corrects errors missed by DNA polymerase. Prokaryotes, MUTS detects mismatch MUDL, and MUTH cleaves incorrect strand. Eukaryotes, MSH2, MLH1, PMS2. 4.3. DNA damage repair mechanisms base excision repair BER corrects minor base modifications. Nucleotide excision repair NER removes bulky DNA lesions. Double strand break DSB repair, homologous recombination HR error-free. Non-homologous end joining NHEJ error-prone. 4.4. Role of telomerase in replication fidelity prevents loss of genetic information at chromosome ends. Eukaryotic linear chromosomes require telomerase to extend telomeres. Practice questions with answers. Previous year question, Gate ecology and evolution. Q1. Which of the following DNA polymerases is responsible for leading strand synthesis in eukaryotes? A. DNA polymerase alpha. B. DNA polymerase delta. C. DNA polymerase epsilon. D. DNA polymerase 3. Answer. C. DNA polymerase epsilon. DNA pol epsilon synthesizes the leading strand, while DNA pol delta synthesizes the lagging strand. Multiple choice questions MCQs Q2. What is the function of DNA helicase in replication? A. Synthesizing RNA primers. B. Relieving supercoiling. C. Unwinding the DNA double helix. D. Proofreading the newly synthesized strand. Answer. C. Unwinding the DNA double helix. Helicase breaks hydrogen bonds between base pairs to separate the strands. Q3. Which of the following is not a function of DNA ligase? A. Sealing nicks between Okazaki fragments. B. Adding nucleotides to the leading strand. C. Forming phosphodiester bonds. D. Connecting repaired DNA segments. Answer. B. Adding nucleotides to the leading strand. DNA ligase does not synthesize DNA, it seals nicks. This detailed explanation covers all major aspects of DNA replication while maintaining a high academic standard for PhD entrance preparation. Let me know if you need further elaboration. You said. Fidelity of replication, extrachromosomal replicons, DNA damage and repair mechanisms, homologous and site-specific recombination, ChatGPT said. DNA replication fidelity, extrachromosomal replicons, DNA damage and repair, and recombination. DNA replication, repair, and recombination are essential processes that maintain genome integrity, prevent mutations, and enable genetic diversity. Below is a detailed explanation of these topics. 1. Fidelity of DNA replication. The fidelity of DNA replication ensures the accurate duplication of genetic material, minimizing mutations. The error rate of replication is approximately 1 error per 10 to the power of 9 10 nucleotides due to several proofreading and repair mechanisms. 1.1. Mechanisms ensuring high fidelity. 1.1.1. Base pair complementarity. DNA polymerases follow Watson-Crick base pairing rules, AT, GC, ensuring accurate incorporation of nucleotides. 1.1.2. Proofreading by DNA polymerases. DNA polymerases have 3 feet right pointing arrow 5, exonuclease activity, allowing them to remove incorrectly incorporated nucleotides. Prokaryotes, DNA pol 3 has proofreading ability. Eukaryotes, DNA pol delta and epsilon have proofreading functions. 1.1.3. Mismatch repair MMR corrects replication errors missed by proofreading. Prokaryotes, MUTS detects mismatches, MIDAL recruits smooth, which cuts the incorrect strand. Eukaryotes, MSH2, MLH1, PMS2 perform similar functions. 1.1.4. DNA damage repair mechanisms. Base excision repair BER corrects minor lesions. Nucleotide excision repair NER removes bulky DNA adducts. Double strand break DSB repair ensures genome stability. 2. Extrachromosomal replicons. Extrachromosomal replicons are DNA elements that replicate independently of chromosomal DNA. These include plasmids, episomes, viral genomes, and mitochondrial DNA. 2.1. Types of extrachromosomal replicons. 2.1.1. Plasmids. Small, circular DNA molecules found in bacteria and some eukaryotes. Carry non-essential genes, including those for antibiotic resistance, are plasmids, or virulence factors. Replicate independently via the theta-theta replication or rolling circle replication mechanism. 2.1.2. Episomes. Plasmids that can integrate into the host genome, e.g., F plasmid and E. coli. Facilitate horizontal gene transfer via conjugation. 2.1.3. Viral genomes. Some viruses maintain extrachromosomal replicons inside host cells, e.g., HIV provirus. Bacteriophage lambda integrates into bacterial DNA as a prophage during lysogeny. 2.1.4. Mitochondrial and chloroplast DNA circular genomes inherited maternally in eukaryotes. Replicate using a replicative DNA polymerase, e.g., POLG in humans. 3. DNA damage and repair mechanisms. DNA is constantly exposed to spontaneous and induced damage, leading to mutations. Cells have evolved repair mechanisms to counteract these errors. 3.1. Types of DNA damage. 3.1.1. Spontaneous damage. Depurination, loss of purine bases A, G. Deamination, cytosine converts to uracil. Tautomeric shifts, base mispairing due to altered hydrogen bonding. 3.1.2. Induced damage. UV radiation, forms pyrimidine dimers, TT or CC dimers. Ionizing radiation, X-rays, gamma rays causes double strand breaks, DSBs. Chemical mutagens, alkylating agents, oxidative damage. 3.2. DNA repair mechanisms. 3.2.1. Direct repair. Photoreactivation, DNA photolase repairs pyrimidine dimers in prokaryotes. Alkyl transferases, e.g., MGMT, remove alkyl groups from guanine. 3.2.2. Excision repair. Base excision repair, BER, repairs single base modifications. DNA glycosylase removes the altered base. AP endonuclease cuts the DNA backbone. DNA polymerase beta eukaryotes or prokaryotes fills the gap. Nucleotide excision repair, NER, repairs bulky lesions. Prokaryotes, UVR ABC endonuclease. Eukaryotes, XPC, XPA, XPB, XPD recognize damage and excise it. 3.2.3. Mismatch repair, MMR. Recognizes and fixes replication errors. MUTS move prokaryotes and MSH2 MLH1 PMS2 eukaryotes. 3.2.4. Double strand break DSB repair homologous recombination HR error-free, uses sister chromatid as a template. Non-homologous end joining NHEJ error-prone, directly joins broken ends. 4. Homologous and site-specific recombination. Recombination is the exchange of genetic material between DNA molecules and plays a crucial role in genetic diversity, repair, and evolution. 4.1. Homologous recombination HR. Meiosis, mitosis DSB repair and horizontal gene transfer. Requires long sequence homology. Key steps. Strand invasion. RADCUP prokaryotes or RAD51 eukaryotes binds to the single-stranded DNA and searches for homologous sequences. Holiday junction formation. DNA strands exchange between homologous chromosomes. Branch migration. The junction moves along the DNA. Resolution. Holiday junctions are cleaved by resolvases. Can result in crossover genetic exchange or non-crossover gene conversion. Examples. Meiotic recombination crossing over. DSB repair by HR BRCA1 and BRCA2 in eukaryotes. 4.2. Site-specific recombination. Recombination occurs at specific DNA sequences, mediated by recombinases. Does not require long sequence homology. Key enzymes. Integrases, e.g., lambda integrase in bacteriophages. Cre-recombinase used in genetic engineering. Transposases for transposon movement. Examples. Integration of bacteriophage lambda into E. coli genome. VDJ recombination in immunoglobulin genes. Cre-LOX-P system for conditional gene knockout. Practice questions with answers. Previous year question. Gate ecology and evolution. Q1. Which DNA repair mechanism is responsible for fixing UV-induced thymine dimers? A. Base excision repair. B. Nucleotide excision repair. C. Non-homologous end joining. D. Mismatch repair. Answer. B. Nucleotide excision repair. NER removes bulky DNA lesions such as thymine dimers. Multiple choice questions. MCQs. Q2. What is the function of RECA in homologous recombination? A. DNA polymerization. B. Recognition of mismatch repair sites. C. Strand invasion and homologous pairing. D. Cleaving phosphodiester bonds. Answer. C. Strand invasion and homologous pairing. Q3. Which enzyme is responsible for resolving holiday junctions? A. DNA polymerase. B. Ligase. C. Resolvase. D. Topoisomerase. Answer. C. Resolvase. This high-level PhD entrance preparation note comprehensively covers replication fidelity, extrachromosomal replicons, DNA repair, and recombination mechanisms with previous year questions and MCQs for effective revision. Let me know if you need further clarification. You said. RNA synthesis and processing, transcription factors and machinery. ChatGPT said. RNA synthesis and processing, transcription factors and machinery. RNA synthesis, or transcription, is the process by which genetic information from DNA is copied into RNA by RNA polymerase. In eukaryotes and prokaryotes, transcription involves various transcription factors, regulatory elements, and enzymes that ensure accurate gene expression. 1. Overview of transcription. Transcription occurs in 3 phases. Initiation, RNA polymerase binds to the promoter region with the help of transcription factors. Elongation, RNA polymerase synthesizes RNA by complementary base pairing with the DNA template strand. Termination, transcription stops, and the RNA molecule is released. In prokaryotes, transcription occurs in the cytoplasm, while in eukaryotes, it takes place in the nucleus before RNA is processed and exported to the cytoplasm. 2. Transcription machinery in prokaryotes. 2.1. RNA polymerase in prokaryotes. Prokaryotes have a single RNA polymerase enzyme that transcribes all types of RNA, mRNA, rRNA, and tRNA. This enzyme consists of core enzyme alpha BB, omega responsible for RNA synthesis. Sigma factor sigma recognizes the promoter and initiates transcription. 2.2. Transcription factors in prokaryotes. Sigma factors different sigma factors recognize different promoters. Example, sigma to the power of 7 housekeeping genes, sigma cubed squared heat shock response. Repressors and activators. Lac repressor LAC inhibits transcription of the lac operon in E. coli. CAMP receptor protein CRP enhances transcription when bound to CAMP. 2.3. Promoter elements in prokaryotes. 10 region Pribnow box, TATAAT, and -35 region TTGACA help RNA polymerase bind and initiate transcription. 2.4. Termination in prokaryotes. Rho-dependent termination requires the Rho factor, which dissociates RNA from DNA. Rho-independent termination involves a GC-rich hairpin loop followed by a poly-U sequence. 3. Transcription machinery in eukaryotes. 3.1. RNA polymerases in eukaryotes. Eukaryotes have 3 major RNA polymerases. RNA polymerase I right pointing arrow synthesizes rRNA, 28S, 18S, 5.8S rRNAs. RNA polymerase II right pointing arrow transcribes mRNA and some snRNA. RNA polymerase III right pointing arrow transcribes tRNA, 5S rRNA, and other small RNAs. 3.2. General transcription factors GTFs in eukaryotes. RNA polymerase II requires several general transcription factors to initiate transcription. Factor function. TFIID contains TBP, binds to TATA box and recruits other factors. TFIIB helps RNA pol II position at the promoter. TFIIF stabilizes RNA pol II binding. TFIIE recruits TFIIH. TFIIH helicase activity unwinds DNA and kinase activity phosphorylates RNA pol II CTD. 3.3. Eukaryotic promoter elements. Core promoter. TATA box -25 region found in some genes, binds TFIID. Initiator INR, sequence, alternative to TATA box. BRIEF TFIIB recognition element, located upstream of TATA. DPE downstream promoter element found in TATA-less promoters. Enhancers and silencers. Enhancers, increase transcription by binding activators. Silencers, decrease transcription by binding repressors. 3.4. Termination in eukaryotes. RNA polymerase II termination involves cleavage of the RNA transcript and polyadenylation. RNA polymerase I and III terminate via different mechanisms involving specific termination signals. 4. RNA processing in eukaryotes. After transcription, primary RNA transcripts undergo several modifications to become mature RNA. 4.1. mRNA processing in eukaryotes. 5' capping. Addition of a 7-methylguanosine M to the power of 7G cap at the 5' end. Helps with mRNA stability and ribosome recognition. Splicing. Removal of introns non-coding regions and joining of exons coding regions. Spliceosome complex SNRNPs, U1, U2, U4, U5, U6 facilitates splicing. Polyadenylation. Addition of a poly A tail approximately 200 adenines at the 3' end. Protects mRNA from degradation and aids translation. RNA editing. Some mRNAs undergo base modifications, e.g., 8I editing adenosine to inosine in neurotransmitter receptors. 4.2. rRNA and tRNA processing. rRNA processing. Precursor 45S rRNA is cleaved into 18S, 28S, and 5.8S rRNAs. tRNA processing. Trimming of 5' and 3' ends. Addition of CCA sequence at 3' end. Base modifications e.g., pseudouridine, inosine. 5. Regulation of transcription. 5.1. Epigenetic regulation. Histone modifications. Acetylation activates transcription. Methylation can activate or repress. DNA methylation. CpG island methylation silences genes. 5.2. Transcriptional activators and repressors. Activators bind enhancers to promote transcription. Repressors bind silencers to inhibit transcription. 5.3. Chromatin remodeling complexes SWI SNF complex opens chromatin for transcription. Polycomb group proteins silence genes. Practice questions with answers. Previous year question. Gate ecology and evolution. Q1. Which of the following is a function of the TATA binding protein TBP? A. Termination of transcription. B. RNA splicing. C. Recognition of the TATA box in promoters. D. Translation initiation. Answer. C. Recognition of the TATA box in promoters. Multiple choice questions. MCQs. Q2. Which RNA polymerase is responsible for synthesizing tRNA in eukaryotes? A. RNA polymerase I. B. RNA polymerase II. C. RNA polymerase III. D. RNA polymerase IV. Answer. C. RNA polymerase III. Q3. Which transcription factor in eukaryotes is responsible for unwinding DNA at the promoter? A. TFIIB. B. TFIIE. C. TFIIH. D. TFIID. Answer. C. TFIIH. This detailed PhD entrance preparation note covers transcription factors, RNA synthesis machinery, and RNA processing with previous year questions and MCQs for revision. Let me know if you need any further clarifications. You said, formation of initiation complex, transcription activator and repressor, RNA polymerases, capping elongation, and termination. ChatGPT said, formation of initiation complex, transcription activators and repressors, RNA polymerases, capping, elongation, and termination. 1. Formation of the initiation complex. 1.1. Prokaryotic transcription initiation complex in prokaryotes, RNA polymerase directly binds to the promoter with the help of sigma factors. Steps. RNA polymerase holoenzyme formation. The core enzyme alpha BB, omega associates with a sigma factor to form the holoenzyme. Promoter recognition. The sigma factor recognizes the -10 Pribnow box, TATAAT, and -35 TTGACA sequences. Closed complex formation. The RNA polymerase holoenzyme binds to the promoter but the DNA remains double-stranded. Open complex formation. RNA polymerase unwinds approximately 12 to 14 bp of DNA near the transcription start site +1. Abort of initiation. Short RNA sequences, approximately 10 nucleotides, are synthesized and released. Promoter clearance. Once RNA synthesis stabilizes, approximately 10 nucleotides long, sigma factor dissociates, and RNA polymerase transitions to elongation. 1.2. Eukaryotic transcription initiation complex. In eukaryotes, transcription initiation is more complex, requiring multiple general transcription factors (GTFs). Steps. Pre-initiation complex formation. TFIID containing TBP, tau binding protein, binds to the tau box -25 region. TFIIB recognizes the Bri element and helps recruit RNA polymerase II. TFIIF stabilizes RNA pol II binding to the promoter. TFIIE and TFIIH with helicase and kinase activity are recruited. DNA unwinding. TFIIH helicase activity unwinds DNA, forming the open complex. TFIIH kinase activity phosphorylates the C-terminal domain (CTD) of RNA pol II, triggering promoter clearance. Promoter clearance and transition to elongation. RNA pol II detaches from general transcription factors and starts elongation. 2. Transcription activators and repressors 2.1. Transcription activators. Definition: proteins that enhance transcription by binding to enhancers or interacting with transcription machinery. Mechanism of action. Recruit RNA polymerase and transcription factors. Open chromatin by recruiting histone acetyl transfer as is PATS. Example: CAM receptor protein (CRP) in prokaryotes. 2.2. Transcription repressors. Definition: proteins that inhibit transcription by blocking activator binding or compacting chromatin. Mechanism of action. Bind to silencers and prevent RNA polymerase binding. Recruit histone deacetylases (HDACs) to close chromatin. Example: lac repressor lacey in E. coli. 3. RNA polymerases. 3.1. Prokaryotic RNA polymerase. A single RNA polymerase transcribes all RNA types. Composed of a core enzyme alpha BB, omega, and a sigma factor for promoter recognition. 3.2. Eukaryotic RNA polymerases. RNA polymerase I transcribes rRNA 28S, 18S, 5.8S rRNA. RNA polymerase II transcribes mRNA, snRNA, mRNA. RNA polymerase III transcribes tRNA, 5S rRNA, and small RNAs. Each polymerase recognizes distinct promoter elements and requires specific transcription factors. 4. Capping of mRNA, eukaryotic only function. Protects mRNA from degradation. Enhances translation initiation process. Addition of a 7-methylguanosine M to the power of 7G cap at the 5' end. Enzymes involved. RNA triphosphatase removes the gamma phosphate. Ganulyltransferase adds GMP in a 5' triphosphate bond. Methyltransferase adds a methyl group to guanine. Prokaryotic mRNA lacks capping and is rapidly degraded after use. 5. Elongation of transcription 5.1. Prokaryotic elongation. After sigma factor dissociation, the core enzyme moves along the DNA, adding ribonucleotides complementary to the template strand. RNA polymerase maintains a 17 bp transcription bubble. 5.2. Eukaryotic elongation. RNA pol II continues transcription while associated with elongation factors. CTD phosphorylation by TFIIH kinase allows recruitment of RNA processing factors, splicing, capping. 6. Termination of transcription. 6.1. Prokaryotic termination. 2 main mechanisms. Rho-dependent termination. Rho factor, an RNA helicase, binds to a rut site on RNA and disrupts RNA DNA binding. Rho-independent termination. Formation of a GC-rich hairpin loop followed by a poly-U stretch leads to RNA detachment. 6.2. Eukaryotic termination. RNA polymerase I uses termination factor TTF1 to stop transcription. RNA polymerase III stops transcription at specific poly-T sequences. RNA polymerase II. Cleavage of the transcript at the polyadenylation signal AAU-AAA. Addition of a poly-A tail by poly-A polymerase for stability. Practice questions with answers. Previous year question: gave ecology and evolution. Q1. Which of the following transcription factors is responsible for unwinding DNA during transcription initiation in eukaryotes? A. TFIID. B. TFIIEC, TFIIHD, TFIIB. Answer: C. TFIIH. Multiple choice questions MCQs. Q2. What is the role of the 7-methylguanosine cap in eukaryotic mRNA? A. Prevents ribosome binding. B. Facilitates mRNA degradation. C. Enhances translation and stability. D. Blocks RNA polymerase binding. Answer: C. Enhances translation and stability. Q3. Which enzyme is responsible for adding the 5' cap to eukaryotic mRNA? A. RNA polymerase I. B. RNA polymerase II. C. Ganulyltransferase. D. Poly-A polymerase. Answer: C. Ganulyltransferase. This detailed PhD entrance study note covers transcription initiation, RNA polymerases, activators, repressors, elongation, capping, and termination with previous year and practice questions. Let me know if you need more explanations or specific topics. You said RNA processing, RNA editing, splicing, and polyadenylation, structure and function of different types of RNA, RNA transport. ChatGPT said RNA processing, editing, splicing, polyadenylation, structure and function of different RNAs, and RNA transport. 1. RNA processing. RNA processing involves modifications to the primary RNA transcript to produce functional RNA molecules. These modifications include capping, splicing, polyadenylation, and editing. Prokaryotes, mRNA is mostly unprocessed and translated immediately. Eukaryotes, pre-mRNA undergoes extensive processing in the nucleus before being transported to the cytoplasm for translation. 2. RNA editing. RNA editing alters the nucleotide sequence of RNA after transcription. This process can generate different protein isoforms from a single gene. 2.1. Types of RNA editing. Substitution editing. Adenosine to inosine ADI editing, catalyzed by adenosine deaminases acting on RNA ADARs. Alters codon specificity, affecting protein function. Cytosine to uracil C2U editing, catalyzed by cytidine deaminases. Example: apolipoprotein B, ApoB mRNA editing in humans, which produces 2 different proteins. Insertion deletion editing. Mediated by guide RNAs GRNAs, common in mitochondrial RNA of trypanosomes. 3. RNA splicing. RNA splicing removes non-coding introns and joins exons to produce a mature mRNA transcript. 3.1. Types of splicing. Spliceosome-mediated splicing, pre-mRNA splicing. Occurs in eukaryotic nuclei and involves small nuclear RNAs, snRNAs, forming a spliceosome complex. Steps of splicing. Recognition of the 5' splice site, GU, and 3' splice site, AG. Formation of a lariat structure at the branch point, a residue. Exon ligation and intron removal. Self-splicing, ribozyme-mediated splicing. Group 1 introns, require GTP as a cofactor. Group 2 introns, form a lariat similar to spliceosomal splicing. Alternative splicing. Generates multiple protein isoforms from a single gene. Examples. Fibronectin gene, different splice variants function in different tissues. Drosophila discam gene, generates thousands of protein isoforms. 4. Polyadenylation. Polyadenylation adds a poly A tail to the 3' end of eukaryotic mRNA to enhance stability and translation. 4.1. Process of polyadenylation cleavage at the polyadenylation signal AAU-AAA. Addition of approximately 200 adenine residues by poly A polymerase PAP. Binding of poly A binding proteins PABPs for stability and translation efficiency. 4.2. Functions of the poly A tail. Enhances mRNA stability, prevents degradation. Aids in nuclear export. Facilitates translation initiation by interacting with the 5' cap. 5. Structure and function of different types of RNA. 5.1. Messenger RNA, mRNA. Structure, linear, single-stranded with a 5' cap and 3' poly A tail, eukaryotes only. Function, carries genetic information from DNA to ribosomes for translation. Prokaryotic versus eukaryotic mRNA. Prokaryotic mRNA, polycistronic, encodes multiple proteins. Eukaryotic mRNA, monocistronic, one gene per mRNA. 5.2. Ribosomal RNA, rRNA. Structure, forms complex secondary structures in ribosomes. Function, catalyzes peptide bond formation during translation, ribozyme activity. Types. Prokaryotic rRNA, 16S small subunit, 23S, 5S large subunit. Eukaryotic rRNA, 18S small subunit, 28S, 5.8S, 5S large subunit. 5.3. Transfer RNA, tRNA. Structure, cloverleaf shape with anticodon and amino acid attachment site. Function, transfers amino acids to ribosomes during translation. Post-transcriptional modifications, pseudouridine, inosine. 5.4. Small nuclear RNA, snRNA. Function, forms the spliceosome, involved in RNA splicing. Examples, U1, U2, U4, U5, U6 snRNAs. 5.5. Small nucleolar RNA, SNO-RNA. Function, guides rRNA modifications, methylation and pseudourylation. Example, C-D-box SNO-RNAs modify ribosomal RNA. 5.6. MicroRNA, mRNA, and small interfering RNA, siRNA, mRNA, regulates gene expression by blocking translation. siRNA, degrades specific mRNAs via RNA interference RNAi. 6. RNA transport. After processing, RNA must be transported to specific cellular locations for function. 6.1. Nuclear export of mRNA mediated by nuclear pore complexes, NPCs. Requires export factors, e.g., NXF1 NXT1 complex in mammals. Export is coupled with splicing and polyadenylation. 6.2. tRNA and rRNA transport. tRNA, processed in the nucleus and exported by exportin T. rRNA, assembled into ribosomal subunits in the nucleolus, then exported to the cytoplasm. Practice questions with answers. Previous year question, Gate Ecology and Evolution, Q1, which RNA processing event involves the removal of introns and joining of exons? A. Capping. B. Splicing. C. Polyadenylation. D. RNA editing. Answer, B. Splicing. Multiple choice questions, MCQs. Q2, which enzyme catalyzes the addition of the 3' poly A tail in eukaryotic mRNA? A. RNA polymerase 2. B. Guanylyltransferase. C. Poly A polymerase D. ADAR. Answer, C. Poly A polymerase. Q3, which RNA molecule has catalytic activity in the ribosome? A. tRNA. B. mRNA. C. rRNA. D. snRNA. Answer, C. rRNA. This detailed PhD entrance study note covers RNA processing, RNA editing, splicing, polyadenylation, RNA structure and function, and RNA transport, along with previous year and practice questions. Let me know if you need any further clarifications. You said. Protein synthesis and processing, ribosome, formation of initiation complex, initiation factors and their regulation, elongation and elongation factors. ChatGPT said. Protein synthesis and processing 1. Ribosome, structure and function 1.1. Ribosome composition. Ribosomes are macromolecular machines that translate mRNA into proteins. They consist of ribosomal RNA, rRNA, and ribosomal proteins. Prokaryotic ribosome, 70S, small subunit 30S, 16S rRNA plus proteins, large subunit 50S, 23S and 5S rRNA plus proteins, eukaryotic ribosome, 80S, small subunit 40S, 18S rRNA plus proteins, large subunit 60S, 28S, 5.8S, and 5S rRNA plus proteins, 1.2. Ribosome function. The ribosome decodes mRNA and catalyzes peptide bond formation. Key sites. A site aminoacyl site binds incoming tRNA with an amino acid. P site peptidyl site holds the growing polypeptide chain. E site exit site where uncharged tRNA exits the ribosome. 2. Formation of the initiation complex 2.1. Prokaryotic initiation complex Shine-Dalgarno sequence. Located upstream of the start codon, August. Recognized by 16S rRNA of the 30S subunit. Steps. 30S ribosomal subunit binding. Initiation factor 3, IF3, prevents premature 50S binding. Initiation factor 1, IF1, blocks the A site. mRNA and FMET tRNA binding. IF2 GTP binds FMET tRNA FMET to the P site. 50S subunit assembly. GTP hydrolysis releases If1, IF2, and IF3. Functional 70S ribosome is assembled. 2.2. Eukaryotic initiation complex Kozak sequence. Helps position the ribosome at the correct August start codon. Steps. 40S subunit binds initiation factors EIFs, EIF1, EIF1A, and EIF3 keep the 40S subunit dissociated from 60S. EIF2 GTP binds MET tRNA MET and brings it to the P site. mRNA recognition and scanning. EIF4 complex EIF4E, EIF4G, EIF4A, EIF4B binds the 5' cap and unwinds secondary structures. The 40S subunit scans for the August start codon. 60S subunit joining. EIF5B GTP hydrolysis triggers release of EIFs. Functional ADS ribosome is assembled. 3. Initiation factors and their regulation 3.1. Prokaryotic initiation factors if 1. Blocks the A site until the ribosome is ready for elongation. IF2. GTP binding protein that helps FMET tRNA bind to the ribosome. If 3. Prevents premature binding of the 50S subunit. 3.2. Eukaryotic initiation factors EIF2. Brings MET tRNAi to the ribosome. EIF4E. Binds the 5' cap of mRNA. EIF4G. Scaffolding protein that bridges EIF4E and other factors. EIF5B. Facilitates 60S subunit joining. Regulation. Phosphorylation of EIF2 alpha inhibits translation during stress conditions. mTOR signaling pathway regulates EIF4E availability, controlling protein synthesis rates. 4. Elongation of translation. 4.1. Steps of elongation aminoacyl tRNA entry. Elongation factor 2 EF2 GTP binds aminoacyl tRNA and delivers it to the A site. GTP hydrolysis releases EF2. Peptide bond formation. Peptidyl transferase 23 srRNA in prokaryotes, 28 srRNA in eukaryotes catalyzes bond formation. Peptide chain moves from P site tRNA to A site tRNA. Translocation. Elongation factor G EFG prokaryotes EF2 eukaryotes hydrolyzes GTP to shift the ribosome forward by 1 codon. The uncharged tRNA moves to the E site and exits. 5. Elongation factors and their regulation 5.1. Prokaryotic elongation factors EF2 brings aminoacyl tRNA to the A site. EFTs regenerates EF2 GTP. EFG promotes ribosome translocation. 5.2. Eukaryotic elongation factors EF1A, analogous to EF2, delivers aminoacyl tRNA. EF1B regenerates EF1A GTP. EF2 promotes ribosome translocation. Regulation. Diphtheria toxin and ricin inhibit EF2, blocking protein synthesis. Practice questions with answers. Previous year question: Gait ecology and evolution. Q1. Which elongation factor is responsible for delivering aminoacyl tRNA to the ribosome in prokaryotes? A. EFG. B. EF2C EFTs D. EIF2. Answer: B. EF2. Multiple choice questions MCQs Q2. What is the function of the Shine-Dalgarno sequence? A. Signals termination of translation D. Recruits ribosomes to prokaryotic mRNA C. Facilitates polyadenylation D. Directs mRNA splicing. Answer: B. Recruits ribosomes to prokaryotic mRNA. Q3. Which initiation factor is responsible for preventing premature binding of the large ribosomal subunit in prokaryotes? A. IF1. F1. B. IF2C IF3D EIF4E. Answer: C. IF3. This detailed PhD entrance study note covers ribosome structure, initiation complex formation, initiation factors, elongation, and elongation factors, along with previous year and practice questions. Let me know if you need any further clarifications. You said. Termination, genetic code. Aminoacylation of tRNA, tRNA identity, aminoacyl tRNA synthetase, ChatGPT said. Protein synthesis, termination, genetic code, and tRNA aminoacylation 1. Termination of translation. 1.1. Signals for termination. Translation ends when the ribosome encounters a stop codon in the mRNA. There are 3 stop codons. UAA, ochre, UAG, amber, UGA, opal. These codons do not code for any amino acid and are recognized by release factors RFs instead of tRNAs. 1.2. Termination process recognition of the stop codon. Prokaryotes. Release factor 1 RF1 recognizes UAA and UAG. Release factor 2 RF2 recognizes UAA and UGA. Release factor 3 RF3 GTPase that facilitates RF1 RF2 activity. Eukaryotes. ERF1 recognizes all 3 stop codons. ERF3 GTPase that enhances ERF1 function. Hydrolysis of the peptidyl tRNA bond. The peptide chain is released from the tRNA in the P site. Dissociation of the ribosome. Ribosomal subunits, mRNA, and release factors disassemble for future translation rounds. Regulation. Nonsense-mediated decay NMD degrades mRNAs with premature stop codons to prevent production of faulty proteins. 2. The genetic code. 2.1. Features of the genetic code. Triplet code. Each amino acid is specified by a codon of 3 nucleotides. Degeneracy. Multiple codons can encode the same amino acid. Universal. The same codons specify the same amino acids across almost all organisms. Non-overlapping. Each nucleotide is part of only one codon. Unambiguous. Each codon codes for only one amino acid. Wobble hypothesis. The third base of a codon can tolerate mismatches, allowing flexibility in tRNA binding. Example: GU wobble pairing. 2.2. Start and stop codons. Start codon: AUG methionine in eukaryotes, N-formylmethionine in prokaryotes. Stop codons: UAA, UAG, UGA. 2.3. Exceptions to the universal code. Mitochondria have slight variations, e.g., UGA codes for tryptophan instead of a stop codon in mammalian mitochondria. Some protists use alternative codons. 3. Aminoacylation of tRNA, tRNA charging. 3.1. Overview. Aminoacylation or charging attaches an amino acid to its corresponding tRNA, forming aminoacyl tRNA. This process is catalyzed by aminoacyl tRNA synthetases, AARSs. 3.2. Steps of aminoacylation: activation of amino acid. The amino acid reacts with ATP, forming aminoacyl AMP. Enzyme: aminoacyl tRNA synthetase. Reaction: amino acid plus ATP right pointing arrow. Aminoacyl AMP plus PPI. Amino acid plus ATP right pointing arrow. Aminoacyl AMP plus PPI. Transfer to tRNA. The aminoacyl group is transferred to the 3'OH CCA tail of tRNA. Reaction: aminoacyl AMP plus tRNA right pointing arrow. Aminoacyl tRNA plus AMP. Aminoacyl AMP plus tRNA right pointing arrow aminoacyl tRNA plus AMP 3.3. Function of aminoacyl tRNA carries the correct amino acid to the ribosome. Forms peptide bonds with the growing polypeptide chain. 4. tRNA identity and recognition 4.1. Structure of tRNA. tRNAs have a cloverleaf secondary structure and an L-shaped tertiary structure. Key regions include anticodon loop, recognizes the mRNA codon. Acceptor stem 3, CCA tail binds the amino acid. D loop and T-C loop, provide structural stability and interact with ribosomal component. 4.2. Determinants of tRNA identity. Anticodon sequence, recognizes the correct codon in mRNA. Acceptor stem nucleotides, specific sequences help aminoacyl tRNA synthetases recognize the correct tRNA. Post-transcriptional modifications, example, inosine I in the anticodon enhances wobble pairing. 5. Aminoacyl tRNA synthetases AARSs. 5.1. Types of aminoacyl tRNA synthetases class 1. Attaches the amino acid to the 2-pheto of the ribose, later shifts to 3-pheto. Example, glutamyl tRNA synthetase. Class 2. Directly attaches the amino acid to the 3-pheto of ribose. Example, serine tRNA synthetase. 5.2. Fidelity of aminoacyl tRNA synthetase is highly specific for both amino acid and tRNA. Some enzymes have proofreading activity. If incorrect amino acid is attached, the enzyme hydrolyzes the bond, preventing errors in translation. Example, isoleucyl tRNA synthetase rejects mischarged valine. Practice questions with answers. Previous year question, Gate Ecology and Evolution, Q1. Which of the following recognizes stop codons during translation in prokaryotes? A. EFG. B. RF1 and RF2. C. ERF1. D. IF3. Answer, B. RF1 and RF2. Multiple choice questions, MCQs. Q2. Which enzyme is responsible for charging tRNA with an amino acid? A. RNA polymerase. B. Peptidyltransferase. C. Aminoacyl tRNA synthetase. D. EF2. Answer, C. Aminoacyl tRNA synthetase. Q3. What is the function of the genetic code's degeneracy? A. Allows multiple amino acids to be encoded by a single codon. B. Enables a single amino acid to be encoded by multiple codons. C. Prevents translation errors. D. Facilitates ribosome recycling. Answer, B. Enables a single amino acid to be encoded by multiple codons. This comprehensive PhD entrance study note covers translation termination, genetic code, tRNA aminoacylation, tRNA identity, and aminoacyl tRNA synthetases, along with previous year and practice questions. Let me know if you need further clarifications. You said, and translational. Proofreading, translational inhibitors, post-translational modification of proteins. ChatGPT said. Protein synthesis, translational proofreading, inhibitors, and post-translational modifications 1. Translational proofreading. 1.1. Importance of translational accuracy. Translational proofreading ensures that the correct amino acids are incorporated into the growing polypeptide chain. Errors in translation can lead to non-functional or toxic proteins. 1.2. Mechanisms of translational proofreading 1.2.1. Proofreading by aminoacyl tRNA synthetases editing site in synthetases. Some aminoacyl tRNA synthetases have an editing domain that hydrolyzes incorrectly charged amino acids. Example, isoleucyl tRNA synthetase rejects mischarged valine. 1.2.2. Proofreading by ribosome, kinetic proofreading, the ribosome delays peptide bond formation until correct codon-anticodon pairing is verified. Elongation factor 2, EF2, prokaryotes, EF1A, eukaryotes binds aminoacyl tRNA and only releases it if codon-anticodon pairing is correct. Incorrectly bound tRNAs are released before peptide bond formation. 1.2.3. Ribosomal decoding center. The 16 srRNA prokaryotes and 18 srRNA eukaryotes check for correct base pairing. Mismatch recognition triggers GTP hydrolysis delay, allowing incorrect tRNAs to dissociate. 2. Translational inhibitors. 2.1. Antibiotics targeting translation. Prokaryotic translation inhibitors used as antibiotics. Streptomycin binds the 30S subunit, causing misreading of mRNA. Tetracycline prevents tRNA binding to the A site. Chloramphenicol inhibits peptidyltransferase activity of the 50S subunit. Erythromycin binds the 23 srRNA of the 50S subunit, blocking elongation. Puromycin mimics aminoacyl tRNA and causes premature chain termination. Eukaryotic translation inhibitors used in research or toxins. Cyclohexamide inhibits peptidyltransferase in the 60S ribosomal subunit. Diphtheria toxin inactivates EF2, blocking translocation. Ricin cleaves 28 srRNA, preventing translation. 3. Post-translational modification (PTM) of proteins 3.1. Importance of PTMs. Post-translational modifications regulate protein stability, activity, localization, and interactions. 3.2. Types of post-translational modifications 3.2.1. Covalent modifications. Phosphorylation. Addition of phosphate group co-cubed to ester, THR, or TR residues. Enzyme, kinases reversed by phosphatases. Function, regulates signaling pathways e.g., MAPK, insulin signaling. Acetylation. Addition of acetyl group coach to Li residues. Example, histone acetylation regulates gene expression. Methylation. Addition of methyl groups CH to Li or ARG residues. Function, controls epigenetic regulation e.g., histone methylation. Ubiquitination. Addition of ubiquitin to Li residues. Function, marks proteins for proteasomal degradation via the ubiquitin proteasome pathway. Sumolation. Addition of sumo, small ubiquitin-like modifier, to proteins. Function, alters protein localization and stability. 3.2.2. Structural modifications. Disulfide bond formation between cysteine residues to stabilize protein structure. Example, insulin maturation requires disulfide bond formation. Proteolytic cleavage. Proteins are activated by removal of specific segments. Example, proinsulin right pointing arrow insulin after cleavage by proteases. Lipidation, membrane targeting. Myristoylation and palmitoylation, attach fatty acids to proteins for membrane anchoring. Example, ROS protein lipidation is essential for its function in cell signaling. Glycosylation, carbohydrate attachment. N-linked glycosylation, adds sugar to asparagine ASN in the ER. O-linked glycosylation, adds sugar to serine SR or threonine THR. Function, affects protein stability, folding, and immune recognition e.g., blood group antigens. Practice questions with answers. Previous year question Gait Ecology and Evolution Q1. Which of the following antibiotics inhibits peptidyltransferase activity in prokaryotes? A. Tetracycline. B. Streptomycin. C. Chloramphenicol. D. Cyclohexamide. Answer. C. Chloramphenicol. Multiple choice questions MCQs. Q2. Which enzyme removes phosphate groups from phosphorylated proteins? A. Kinase. B. Phosphatase. C. Ubiquitin ligase. D. Peptidyltransferase. Answer. B. Phosphatase. Q3. Ricin inhibits translation by targeting which ribosomal component? A. 30S subunit. B. 50S subunit. C. 28SRRNA. D. EFG. Answer. C. 28SRRNA. This detailed PhD entrance study note covers translational proofreading, inhibitors, and post-translational modifications with previous year and practice questions. Let me know if you need further elaboration. You said. Control of gene expression at transcription and translation level, regulating the expression of phages, viruses. ChatGPT said. Control of gene expression at transcription and translation levels 1. Introduction to gene expression regulation. Gene expression regulation ensures that genes are expressed at the right time, place, and amount in response to environmental and cellular signals. This regulation occurs at transcriptional and translational levels. 1.1. Levels of gene regulation. Transcriptional regulation controls mRNA production. Post-transcriptional regulation includes mRNA splicing, stability, and transport. Translational regulation affects ribosome binding and protein synthesis rate. Post-translational regulation modifies protein function through phosphorylation, ubiquitination, etc. 2. Transcriptional regulation of gene expression. 2.1. Regulation in prokaryotes. 2.1.1. Operon model. Jacob and Monod, 1961. Operons are clusters of functionally related genes regulated together. Types of operons. Inducible operon, e.g., lac operon, activated in the presence of an inducer. Repressible operon, e.g., TRP operon, turned off in the presence of a corepressor. Example, lac operon, inducible system. Regulates lactose metabolism in E. coli. Components. Latch, LACI, lac structural genes encoding beta-galactosidase, Permes, transacylase. LACI repressor gene binds to the operator to inhibit transcription. Inducer allolactose binds to LACI, inactivating repression. Example, TRP operon, repressible system, regulates tryptophan biosynthesis in E. coli. High tryptophan, TRP acts as a corepressor, activating the TRP repressor to block transcription. Low tryptophan, repressor is inactive, allowing gene expression. 2.1.2. Sigma factor regulation. In prokaryotes, different sigma factors control transcription initiation. Example. Sigma to the power of 7, housekeeping genes. Sigma cubed squared, heat shock response genes. 2.2. Regulation in eukaryotes. 2.2.1. Chromatin modifications, epigenetic regulation, histone acetylation. Histone acetyltransferases, HATs, add acetyl groups. Right pointing arrow loosens chromatin. Right pointing arrow activates transcription. Histone deacetylases, HDACs, remove acetyl groups. Right pointing arrow tightens chromatin. Right pointing arrow represses transcription. DNA methylation. Cytosine methylation at CpG islands represses transcription. Example, silencing of tumor suppressor genes in cancer. 2.2.2. Transcription factors, TFs, and enhancers. Transcription factors, TFs, bind to promoter enhancer sequences to regulate RNA polymerase activity. Enhancers, DNA regions that bind activator proteins to increase transcription. Silencers, DNA regions that bind repressors to inhibit transcription. 2.2.3. Hormonal regulation of gene expression. Steroid hormones, e.g., estrogen, testosterone, bind intracellular receptors that act as TFs. Peptide hormones, e.g., insulin, activate cell signaling pathways to regulate TFs. 3. Translational regulation of gene expression. 3.1. Regulation in prokaryotes. Riboswitches. mRNA structures that bind small molecules to regulate translation. Example, thymine riboswitch in bacteria. Small RNAs, sRNAs. Bind to mRNA and block ribosome binding or promote degradation. 3.2. Regulation in eukaryotes. 3.2.1. MicroRNAs, mRNAs, and RNA interference mRNAs bind to mRNA, leading to degradation or translation inhibition. Example, miR21 suppresses tumor suppressor genes in cancer. 3.2.2. Regulation via translation initiation factors. EIF2 phosphorylation inhibits translation initiation during stress conditions. EIF4 E-binding proteins, 4 EBPs, block cap-dependent translation. 4. Regulation of phages and viruses. 4.1. Bacteriophage gene regulation. 4.1.1. Lytic versus lysogenic cycle regulation. Lambda-phage, lytic cycle. Viral genes are transcribed, new phages are produced, and host cells lyse. Crow-protein represses lysogenic genes and activates lytic genes. Lysogenic cycle. Phage DNA integrates into the host genome, prophage state. C repressor protein maintains lysogeny by blocking lytic genes. Induction. Under stress, RECA degrades C, switching to the lytic cycle. 4.2. Viral gene expression regulation. 4.2.1. HIV retrovirus transcriptional regulation. TAT protein enhances RNA polymerase elongation, increasing viral mRNA production. Rev protein regulates nuclear export of unspliced viral RNAs. 4.2.2. SARS-CoV-2 translational regulation. Cap-dependent translation uses host ribosomes for viral protein synthesis. Frame shifting generates multiple proteins from a single RNA transcript. Practice questions with answers. Previous year question Gait Ecology and Evolution Q1. In the lac operon, which molecule acts as an inducer? A. Lactose. B. Glucose. C. Allolactose. D. Camp. Answer. C. Allolactose. Multiple choice questions MCQs. Q2. Which of the following regulates gene expression at the transcriptional level? A. mRNAs. B. Histone acetylation. C. Riboswitches. D. Peptidyltransferase. Answer. B. Histone acetylation. Q3. Which bacterial operon is an example of a repressible system? A. Lac operon. B. TRP operon. C. Arabinose operon. D. Heat shock operon. Answer. B. TRP operon. This detailed PhD entrance study note covers transcriptional and translational regulation of gene expression, including operons, chromatin modifications, small RNAs, and viral gene expression control, along with previous year and practice questions. Let me know if you need further details. You said. Prokaryotic and eukaryotic genes, role of chromatin in gene expression and gene silencing. ChatGPT said. Prokaryotic and eukaryotic genes, role of chromatin in gene expression and gene silencing. 1. Prokaryotic versus eukaryotic genes. 1.1. Differences between prokaryotic and eukaryotic genes. Feature prokaryotic genes. Eukaryotic genes. Organization operon-based, e.g., lack operon, individual genes with regulatory elements, promoter structure simple, 10 Pribnow box and -35 sequences complex, Tau box, GC box, CAAT box, RNA polymerase single, RNA polymerase 3, RNA polymerases, poli, 2, 3 mRNA processing, no introns, no capping or polyadenylation splicing, 5 feed capping, 3 feed polyadenylation, gene regulation primarily at transcriptional level, multiple levels, transcriptional, post-transcriptional, epigenetic. 1.2. Prokaryotic genes. Polycistronic mRNA, a single mRNA can encode multiple proteins, e.g., lack operon. Regulation, operons controlled by repressors, activators, and inducers. 1.3. Eukaryotic genes. Monocistronic mRNA, one gene right pointing arrow, one mRNA right pointing arrow, one protein. Enhancers and silencers, DNA regions regulate transcription from a distance. 2. Role of chromatin in gene expression and gene silencing. 2.1. Chromatin structure and gene regulation. 2.1.1. Nucleosome composition. DNA wraps around histone octamers, H2A, H2B, H3, H4. Linker histone H1 stabilizes chromatin compaction. 2.1.2. Chromatin states. Euchromatin, active chromatin, loosely packed, transcriptionally active. Heterochromatin and active chromatin, densely packed, transcriptionally silent. Constitutive heterochromatin, always silent, e.g., centromeres, telomeres. Facultative heterochromatin, can switch between active and inactive states, e.g., X inactivation in females. 2.2. Epigenetic modifications and gene regulation. 2.2.1. Histone modifications, histone acetylation, activation. Histone acetyltransferases, HATs, add acetyl groups, right pointing arrow loosens chromatin, right pointing arrow increases transcription. Histone deacetylases, HDACs, remove acetyl groups, right pointing arrow compacts chromatin, right pointing arrow represses transcription. Histone methylation, activation or repression. H3K4Me3, triangulation at lysine 4 on H3, activates transcription. H3K9Me3 and H3K27Me3, induces gene silencing. 2.2.2. DNA methylation, gene silencing. Addition of methyl CH groups to cytosine, CpG sites, represses transcription. Enzyme, DNA methyltransferases, DNMTs. Example, X chromosome inactivation in females. 2.2.3. Non-coding RNAs, ncRNAs, in gene silencing microRNAs, mRNAs bind mRNA, leading to degradation or translation inhibition. CRNAs, small interfering RNAs, target specific mRNAs for degradation. LNCRNAs, long non-coding RNAs, recruit chromatin modifiers for gene silencing. 3. Gene silencing mechanisms. 3.1. X chromosome inactivation, dosage compensation in females. 1 X chromosome is randomly inactivated in female mammals. Cyst RNA coats the X chromosome, right pointing arrow recruits repressive histone modifiers, right pointing arrow silences genes. 3.2. Imprinting parent-specific gene expression, some genes are expressed only from the maternal or paternal allele. Example, IGF2 paternal and H19 maternal imprinting in mammals. 3.3. RNA interference, RNAi, and gene silencing CRNAs and mRNAs silence genes by degrading target mRNAs. Used in antiviral defense and gene knockdown studies. Practice questions with answers. Previous year question, Gate Ecology and Evolution, Q1, which histone modification is commonly associated with active gene transcription? A. H3K9Me3. B. H3K4Me3. C. DNA methylation. D. H3K27Me3. Answer, B. H3K4Me3. Multiple choice questions, MCQs. Q2, which enzyme catalyzes the addition of acetyl groups to histones? A. Histone deacetylase, HDAC. B. Histone methyltransferase. C. DNA methyltransferase. D. Histone acetyltransferase, HAT. Answer, D. Histone acetyltransferase, HAT. Q3, what is the function of cyst RNA? A. Transcription activation. B. X chromosome inactivation. C. Ribosomal RNA processing. D. Enhancer activation. Answer, B. X chromosome inactivation. This detailed PhD entrance study note covers prokaryotic and eukaryotic genes, chromatin modifications, and gene silencing, with practice questions and answers. Let me know if you need further elaboration. You said. Recombinant DNA methods, isolation and purification of RNA, ChatGPT said. Recombinant DNA methods, isolation and purification of RNA. 1. Introduction to RNA isolation and purification. RNA isolation is a crucial step in molecular biology, enabling gene expression studies, cloning, and recombinant DNA technologies. Due to its instability and susceptibility to degradation by RNases, ribonucleases, RNA isolation requires careful handling. 2. Steps in RNA isolation and purification. 2.1. Sample collection and preparation. Use fresh or properly stored frozen and liquid nitrogen or unrelated biological samples. Work in an RNase-free environment using RNase-free tips, tubes, and gloves. 2.2. RNA isolation methods. 2.2.1. Organic extraction phenol chloroform method. Trizol method uses acidic phenol and guanidine thiocyanate to lyse cells and inhibit RNases. Steps. Cell lysis. Trizol or phenol chloroform disrupts membranes. Phase separation. Centrifugation separates RNA, aqueous phase, DNA, interphase, and proteins, organic phase. RNA precipitation. Isopropanol or ethanol is used to precipitate RNA. RNA washing. RNA pellet is washed with 70% ethanol. Resuspension. RNA is dissolved in RNase-free water. Advantages. High yield, effective RNase inhibition. Disadvantages. Use of hazardous organic solvents, labor-intensive. 2.2.2. Column-based RNA purification. Silica membrane or spin column kits uses silica membranes to selectively bind RNA while removing contaminants. Steps. Lysis and homogenization. Lysis buffer with chiotropic salts disrupts cells. RNA binding. RNA binds to a silica column under high salt conditions. Washing. Impurities, DNA, proteins are removed with ethanol-based buffers. Elution. Pure RNA is eluted in RNase-free water. Advantages. Quick, efficient, no organic solvents. Disadvantages. May yield lower RNA amounts compared to organic extraction. 2.2.3. Magnetic bead-based RNA isolation. Uses oligo-DT beads to capture polyadenylated mRNA or silica-coated magnetic beads. Used in mRNA purification and automated high-throughput RNA isolation. Advantages. High specificity, scalable for automation. Disadvantages. Requires specialized equipment. 2.3. RNA purification and quality assessment 2.3.1. DNA's treatment Y. To remove contaminating DNA. Method. DNA's digestion followed by column purification. 2.3.2. RNA quality and integrity check spectrophotometry nanodrop A260 A280 ratio approximately 2.0 indicates RNA purity. A260 A230 ratio in GT 2.0 indicates low contamination salts phenol. Agarose gel electrophoresis. Intact total RNA shows 28S and 18S rRNA bands 2:1 ratio. Bioanalyzer Agilent RNA integrity number RIN and GT 7 indicates high quality RNA. 3. Applications of purified RNA in recombinant DNA technology cDNA library construction, reverse transcription of mRNA into cDNA for cloning. QRT-PCR quantitative real-time PCR gene expression analysis. RNA-seq transcriptome analysis for functional genomics. In vitro transcription, synthesis of RNA probes for hybridization studies. Practice questions with answers. Previous year question 8 Ecology and evolution Q1. In RNA isolation using the Trizzle method, which phase contains RNA after centrifugation? A. Organic phase. B. Aqueous phase. C. Interphase. D. Pellet. Answer. B. Aqueous phase. Multiple choice questions MCQs Q2. What is the purpose of using DNase in RNA purification? A. To degrade proteins. B. To remove genomic DNA contamination. C. To break down RNA. D. To increase RNA stability. Answer. B. To remove genomic DNA contamination. Q3. What is the ideal A260 A280 ratio for pure RNA? A. 1.5. B. 1.8. C. 2.0. D. 2.5. Answer. C. 2.0. This detailed PhD entrance study note covers RNA isolation and purification techniques, their principles, steps, quality control, and applications in recombinant DNA technology, along with practice questions and answers. Let me know if you need more details. You said, DNA and proteins. Molecular cloning of DNA or RNA fragments in bacterial and eukaryotic systems. ChatGPT said. Molecular cloning of DNA or RNA fragments in bacterial and eukaryotic systems 1. Introduction to molecular cloning. Molecular cloning is a technique used to insert DNA or RNA fragments into a host organism to amplify or express genes of interest. It is widely used in recombinant DNA technology, gene therapy, protein expression, and functional genomics. 2. Steps in molecular cloning 2.1. Preparation of DNA or RNA fragments. Genomic DNA isolation, extracted from cells using phenylchloroform or silica-based methods. mRNA isolation and cDNA synthesis, reverse transcription converts mRNA into complementary DNA, cDNA, for cloning. 2.2. Cutting and processing DNA fragments. Restriction enzyme digestion. Enzymes cut DNA at specific sites to generate sticky or blunt ends. PCR amplification. Polymerase chain reaction (PCR) amplifies the target DNA fragment. cDNA synthesis for RNA cloning. Reverse transcriptase converts RNA into cDNA before cloning. 2.3. Selection of cloning vectors. A vector is a DNA molecule that carries foreign DNA into a host cell. Vector type insert size applications plasmids 1 to 10 kb general cloning, protein expression bacteriophage lambda 9 to 23 kb large DNA fragments cosmids 35 to 45 kb genomic libraries. Bacterial artificial chromosomes 100 to 300 kb large genomic inserts YACs, yeast artificial chromosomes 100 kilobits 1 megabit cloning in eukaryotic systems 2.4. Ligation of DNA into vectors. DNA ligase T4 DNA ligase seals nicks between vector and insert DNA. Ta cloning uses stack polymerase generated A overhangs for ligation. Gibson assembly fuses DNA fragments using exonuclease, polymerase, and ligase. 2.5. Transformation and selection of recombinant clones 2.5.1. Transformation methods in bacteria. Chemical transformation uses calcium chloride CaCl and heat shock to introduce plasmid DNA into E. coli. Electroporation, high voltage electric pulses create membrane pores for DNA entry. 2.5.2. Transfection in eukaryotic cells. Lipofaction, lipid nanoparticles deliver DNA into cells. Electroporation, electric shock allows DNA uptake. Viral transduction, modified viruses introduce genes into mammalian cells. 2.5.3. Selection of recombinants. Antibiotic resistance markers, e.g., ampicillin, amp, kanamycin, con. Blue-white screening, latch, white colonies indicate successful cloning. Fluorescent markers, GFP, green fluorescent clones indicate successful transformation. 3. Cloning in bacterial versus. Eukaryotic systems feature bacterial cloning. Eukaryotic cloning hosts cells E. coli, bacillus yeast, mammalian cells vectors plasmids, phages, BACs YACs, viral vectors expression prokaryotic promoters T7, lac eukaryotic promoters CMV, SV40 post-translational modifications absent present glycosylation, phosphorylation applications recombinant proteins, gene cloning gene therapy, transgenic models 4. Applications of molecular cloning. Recombinant protein production, insulin, growth factors, monoclonal antibodies. Gene therapy, correcting genetic disorders by inserting functional genes. Genomic and cDNA library construction, storing gene collections for research. CRISPR gene editing, inserting or modifying genes in model organisms. Practice questions with answers. Previous year question 8 Ecology and evolution Q1. Which of the following vectors is most suitable for cloning large DNA fragments 100-300 kilobits? A. Plasmid. B. Bacteriophage lambda C. BAC. D. Yak. Answer. C. BAC. Multiple choice questions MCQs Q2. What enzyme is used to ligate DNA fragments into a vector? A. DNA polymerase. B. T4 DNA ligase C. Restriction endonuclease D. Reverse transcriptase. Answer. B. T4 DNA ligase. Q3. What is the purpose of blue-white screening in molecular cloning? A. To check DNA purity. B. To identify recombinant clone. C. To sequence inserted DNA. D. To increase transformation efficiency. Answer. B. To identify recombinant clones. This detailed PhD entrance study note covers molecular cloning steps, bacterial versus eukaryotic systems, and applications, with practice questions and answers. Let me know if you need more details. You said. Expression of recombinant proteins using bacterial, animal and plant vectors, generation of genomic and cDNA libraries in plasmid, phage, cosmid, BAC and YAC vectors, mutagenesis and deletion techniques. ChatGPT said. Expression of recombinant proteins in molecular techniques 1. Expression of recombinant proteins in different systems. Recombinant protein expression involves inserting a gene of interest into a suitable vector and expressing it in a host system. The choice of system depends on factors like post-translational modifications, yield, and cost. 1.1. Expression in bacterial systems common hosts. Escherichia coli, E. coli, Bacillus subtilis vectors, plasmids with strong promoters T7, lac, PET series advantages. High yield, fast growth, cost-effective, easy genetic manipulation disadvantages. Lack of post-translational modifications, glycosylation, phosphorylation, improper protein folding may result in inclusion bodies inducible expression in bacteria T7 promoter system, IPTG induction for high-level expression lac operon system, induced by lactose or IPTG heat shock induction, controlled expression by temperature change 1.2. Expression in animal systems common hosts, mammalian cell lines CHO, HEK293, BHK vectors, viral vectors adenovirus, retrovirus, lentivirus, or plasmid vectors PCMV, PSV40 advantages. Proper post-translational modifications correct protein folding and secretion disadvantages. Expensive culture conditions lower yield compared to bacterial systems 1.3. Expression in plant systems. Common hosts, Nicotiana benthamiana, or Arabidopsis thaliana, Agrobacterium tumefaciens mediated transformation vectors, T plasmid, viral vectors TMV, PVX. Advantages. Cost-effective, large-scale production eukaryotic modifications possible edible vaccines, e.g., recombinant proteins in fruits disadvantages. Lower expression levels compared to bacteria requires stable transformation and selection 2. Generation of genomic and cDNA libraries. A genomic library contains the entire DNA of an organism, while a cDNA library represents only expressed genes, mRNA-derived. 2.1. Construction of genomic library steps. Isolate genomic DNA fragmentation using restriction enzymes insert into vectors plasmids, phages, BACs, YACs transform into host cells applications. Genome sequencing functional genomics 2.2. Construction of cDNA library steps. Isolate mRNA using oligo DT feeds reverse transcription to synthesize cDNA ligate cDNA into vectors transform into host cells applications. Gene expression studies identification of novel transcripts 2.3. Choice of vectors for libraries vector type insert size used for plasmids 1 to 10 kb small genomic cDNA fragments bacteriophage lambda 9 to 23 kb genomic libraries cosmids 35 to 45 kb large DNA fragments. BACs bacterial artificial chromosomes 100 to 300 kb large scale genomic studies YACs yeast artificial chromosomes 100 kilobits 1 megabit eukaryotic genome projects 3. Mutagenesis and deletion techniques 3.1. Site-directed mutagenesis SDM ultra-specific nucleotides in a gene to study protein function. Methods. PCR-based mutagenesis uses mismatched primers to introduce mutations. Oligonucleotide mutagenesis uses synthetic DNA primers with desired changes. Applications. Understanding protein structure function relationships. Developing drug-resistant enzymes 3.2. Random mutagenesis introduces mutations randomly across the genome. Methods. Chemical mutagenesis EMS, ENU induces base pair substitutions. UV mutagenesis induces pyrimidine dimers. Error-prone PCR increases mutation rate in amplified DNA applications. Generating enzyme variants with improved activity. Studying mutational effects in evolution 3.3. Deletion techniques. Remove specific DNA segments to study gene function. Methods. Restriction enzyme-based deletion, cutting and religating DNA CRISPR SOS9 gene knockout, targeted deletion using SOS9 nuclease Krelok system, inducible gene deletions in transgenic organisms. Applications. Functional genomics. Developing knockout models for disease research 4. Practice questions with answers. Previous year question, Gate ecology and evolution Q1, which expression system allows for proper glycosylation of recombinant proteins? A. E. coli. B. Bacillus subtilis. C. Mammalian cells. D. Yeast. Answer, C. Mammalian cells. Multiple choice questions MCQs. Q2, which vector is best suited for constructing a genomic library for large DNA fragments 100-300 kilobits? A. Plasmid. B. Bacteriophage lambda. C. BAC. D. Yak. Answer, C. BAC. Q3, what is the main advantage of using a cDNA library instead of a genomic library? A. Contains only coding sequences, exons. B. Includes regulatory elements and introns. C. More complex than genomic libraries. D. Requires restriction enzyme digestion. Answer, A. Contains only coding sequences, exons. Q4, which technique is used for targeted gene deletion in mammalian cells? A. Error-prone PCR. B. Krelok's recombination. C. Chemical mutagenesis. D. Site-directed mutagenesis. Answer, B. Krelok's recombination. This detailed PhD entrance study note covers recombinant protein expression, genomic and cDNA libraries, mutagenesis, and deletion techniques, along with practice questions and answers. Let me know if you need further elaboration. You said. Gene knockout, DNA and protein sequencing methods. ChatGPT said. Gene knockout and DNA, protein sequencing methods 1. Gene knockout techniques. Gene knockout refers to the deliberate inactivation or deletion of a specific gene to study its function. This technique is commonly used in model organisms, particularly mice, to analyze gene function and develop disease models. 1.1. Homologous recombination. Process, introduces a modified version of a gene into a cell, where it recombines with the endogenous gene, replacing it with a mutant or inactivated form. Used in creating knockout mice and other transgenic animals. Advantages, allows precise genetic modifications. Disadvantages, time-consuming and difficult in certain organisms. Application, generating disease models, studying gene function. 1.2. CRISPR SOS9 technology. Process, a revolutionary genome editing tool that uses the SOS9 enzyme guided by RNA to induce double-strand breaks at specific loci. The resulting repair mechanisms lead to gene knockout by non-homologous end joining, NHEJ, or homology-directed repair, HDR. Advantages, faster, more efficient, and easier than homologous recombination. Disadvantages, off-target effects and incomplete knockouts. Application, knockout of specific genes in various organisms, mice, plants, human cells for functional studies. 1.3. RNA interference, RNAi. Process, involves introducing small interfering RNAs, siRNAs, or short hairpin RNAs, shRNAs, to silence gene expression. Advantages: temporarily silences gene expression without permanent genetic modifications. Disadvantages: incomplete or variable silencing, does not work in all cell types. Application.